We're now going to get into the end, almost the end, with a short video of the adaptive body defenses. This is now going to deal. We're now going to deal here with the cellular immune response, and this involves the T cells, the helper T cells, the cytotoxic T cells. Now, these are the cells that provide defense against intracellular antigens. Examples of this will be cells infected with viruses or bacteria, cancer cells, abnormal cells, or transplanted cells. So T cells directly kill the cells. Okay, they also some of them also would release chemicals that regulate the immune response, but they are very different from the B cells. They're more in that they're more complex than B cells in both their classification and function. And there are two populations of T cells that are based on which cell differentiation receptors that they have on their surface. And the first one is what you call the CD4 cells, and usually they become the helper T cells and the regulatory T cells. And the other one is called the CD8 cells, and they become the cytotoxic T cells. These are the ones that are capable of destroying cells that would carry the foreign antigen. These are the ones that also become the memory T cells. The helper cytotoxic and regulatory T cells are activated T cells. These are also the naive T cells that are simply termed the CD4 or CD8 T cells. So here is a picture, okay, of the T cells, and pretty much they would differentiate. They differentiate, and you could see the difference between the T cell receptors. This is your CD4 cell, and this will be your CD8 cell. And the CD4 are actually destined to become your regulatory T cells right here, and your helper T cells. And the CD8 are destined to become your cytotoxic T cells. So T cells respond only to processed fragments of antigens that are displayed on the surface of cells by MHC proteins. The antigen presentation is vital here for the activation of naive T cells and the normal functioning of your effector T cells. And there are two classes of MHC protein because these are the ones that are presenting. It's just like your MHC protein is the platter by which the antigen is going to be presented to them. You have two types of them. You have two classes, the class 1 MHC protein and the class 2 MHC protein. The class 1 is displayed, you find it in all cells except the red blood cells. In class 2, the MHC proteins are displayed by antigen presenters. These will be your dendritic cells, your macrophages, and both types are synthesized in the endoplasmic reticulum, and they bind to peptide fragments. Let's discuss the MHC1. MHC1 pretty much are very short endogenous antigenic protein, and they can be self-antigen, which means the normal proteins of our cell, or they could be non-self-antigen. These are abnormal proteins that are found in infected or abnormal cells. The class 1, and take note of this, is very important for CD8 activation. The class 1 acts as antigen holders, forming self part that T cells would recognize. So they inform the cytotoxic T cells of microorganisms hiding inside your cells, right? And then your class 2 bind with longer fragments, so they're kind of like bigger, of exogenous extracellular antigens that could have been engulfed or broken down by antigen presenting cells. So the class 2 proteins are recognized by helper T cells. They signal the CD4 cells that help is recognized. So whenever finally the T cells would be bound to a class 2, this is actually going to signal the helper T cells to differentiate. So this is again your cheat sheet, very good display of the MHC1 and MHC2 uh, proteins. and pretty much who recognizes them. This is recognized by the CD8. This is recognized by the CD4, by your class MHC2, and then the type of foreign antigen and the cells displaying foreign antigens on MHC that send this message. Pretty much, this is a very good comparison of the MHC1 and MHC2 protein. If you are, um, if you want, you can actually use this in your review. So there are some restrictions with the major histocompatibility complex. Both the CD4 and the CD8 cells have different requirements for uh, MHC protein that presents antigen to them. And this is important. The CD4 cells that become helper T cells are restricted to binding only to class 2 MHC protein, typically on the antigen presenter surfaces. And your CD8 cells that become cytotoxic T cells are restricted only to binding on class 1 MHC protein found on every cell surface, including your APC cells. But once they are activated, the cytotoxic T cells seek out the same antigen on class 1 MHC proteins on any cell. That is really very important. But there is actually restriction to the MSC. There is a dilemma that the dilemma is actually your antigen presenter. The dilemma for antigen presenter is how to enable the CD8 cells to be activated by non-endogenous phagocytosed proteins. The dendritic cells get around this protein by engulfing dying virus-infected or tumor cells, then displaying the engulfed cells' endogenous protein on both class 1 and class 2 MHC. The dendrites can also import antigens via temporary gap junctions that are infected um, with infected cells rather than engulfing them. So, uh, pretty much, I am going to hold you responsible for reading and knowing the activation and differentiation of T cells from the antigen binding. This is actually where the antigen, the T cell antigen receptors bind to the MHC complex on the APC surface, all the way to co-stimulation. Remember this, guys, that before a T cell can actually act, it has to bind to the antigen. Okay, then it has to be co-stimulated. Who will actually be co-stimulating them? Well, they need to be co-stimulated by, let's say, their something else, by a special chemical. And this is actually the co-stimulation usually is done by a helper T cell. So in the old days, the helper T cell were discovered as helper T cell, but it's not necessarily just helper T cell. These are actually like the big boss. Actually, once your T cell is bound to the antigen, they will have to recognize that that's actually a foreign antigen. And when they recognize it, it's just like a soldier. Oh, I know that you are that you are actually my enemy. I should kill you, but I do not know how to kill you. And this is where the what do you call this? This is where the uh, activation or the co-stimulation of the helper T cell will come into play. Without co-stimulation, anergy occurs in which the T cells will become tolerant to the antigen. They are unable to divide and will not be secreting cytokines. This two-step process would safeguard our bodies against unwanted T cell activation because we do not want any autoimmune response to really happen untowardly. So after the T cell has been co-stimulated, the T cells are now activated and they would enlarge and they will proliferate and they will start to, again, just like your B cell, they will start to proliferate and differentiate. This is where pretty much they will um, clone themselves outside because they already know who to kill and how to kill. They are now what you call activated, and they will start killing any microorganism that would bear the antigen itself. But T cells are hazardous when they are pretty much large in population because they produce large amounts of inflammatory cytokines. The reason why we feel very, very sick is because of these inflammatory cytokines that is actually being produced by T cells in the middle of the battlefield. And they could result in hyperplasia or cancer if they are not treated from the system. And therefore, there is actually like suppressor T cells that would have to make sure that all of the activities of the soldiers of the body will be suppressed if the battlefield is already clear. Memory T cells remain and mediate a secondary response, and that is like they only a small amount of them will have to remain and become memory T cells, and they stay in the lymph node. All right, so here it is showing us pretty much right in this first picture right here. You have an antigen presenter. The dendritic cell, this is your dendritic cell, engulfs an exogenous antigen. And as it engulfs its endogenous oxygen, it processes it and displays the fragments on the class 2 MHC protein. That is actually the class 2 MHC protein, and that's the antigen. The red portion right there in the middle is your antigen. It is now being presented to your naive T cell right here. And there is actually a double recognition here. It's a double recognition because you have the major histocompatibility complex, and it's being presented to that. And what do you call this? The antigen itself being presented. So the CD4 recognizes the antigen and the MHC complex. That's actually two of them. Both, what do you call this? The PCR and the CD4 proteins bind to the MHC complex. And so the co-stimulatory would now begin here. So it is now co-stimulated right here, okay? And the clone formation will start at this point. Clone formation will happen once this particular activated CD4 cells proliferate, okay? And once they realize, once the activation has been realized. So cytokines are special chemical messengers of the immune system. They mediate the development, differentiation, and responses in the immune system. And they include your interferons and interleukins. And they are synthesized by many of our uh, uh, cells. And these are the ones that would activate and help differentiate the T cells. Okay. Example of this will be your gamma interferon. They would enhance the killing power of macrophages. Interleukin 2 is a key growth factor, and they encourage the activated T cell to divide rapidly. And this is actually a list of your cytokine and the function of the cytokine in the immune response. I am not really quite sure if I am inclined to give that to you in your exams. Probably not. So let's take a look at who the big boss really is, the helper T cells. And the helper T cells play a central role in the adaptive immune response. So pretty much both, they activate both humoral and cellular arms. Once primed by the antigen presentation, okay, of our antigen presenters, they help activate the B cells and other T cells. They induce T and B cell proliferation, and they secrete cytokines that recruit other immune cells. Without the helper T cell, there won't be any immune response. And guess what, guys? Once your AIDS virus attack our body, and once we become what do you call this HIV, not only just HIV positive, but we have a full blown AIDS, then our helper T cells are going to be the ones that are attacked, and the entire immune system is going to be compromised because without the helper T cell, there won't be any immune response. So the T cells also activate B cells. Helper T cells interact directly with B cells by displaying antigen fragments bound to MHC2 receptors. And that's exactly pretty much what one of the vital role of the B cell. stimulate. They also stimulate the B cells to provide more rapidly and begin antibody formation. The B cells may be activated without the helper T cells by binding to T cell independent antigen, but the response is weak and very short-lived. So your helper T cell is actually a very good activator. So the central role of the helper T cell, therefore, in mobilizing both is actually to mobilize both the humoral as well as the cellular immunity. It pretty much the helper T cell right here, okay, your helper, your your this is your B cell that is actually being activated. This is your helper T cell. And you can see that the helper T cell binds with the self-clone complexes of a B cell that has encountered its antigen already and displaying its MHC2 on the surface, right? And the helper T cell would release interleukins, and pretty much that will serve as a co-stimulant to pretty much to signal and complete the B cell activation. Okay, here, okay, your helper T cell binds to a dendritic cell. This is your dendritic cell here. And this is actually how helper T cell will help in other cellular immunity. It binds to the dendritic cell, and pretty much the helper T cell stimulates the dendritic cell to express co-stimulatory molecules. That would pretty much like co-stimulate the T cell, and they become sensitized T cells already. So there are other roles of effector T cells. And these are the T cells that are already like cloned and outside already of the blood cells. They amplify responses of the innate immune system. They activate the macrophages, leading to more potent killers. They mobilize lymphocytes and macrophages and attract other types of white blood cells. Now, I am not going to be giving you these subsets, and the reason why I put this in the PowerPoint is so that you know that when you read it in your book, I am not going to give this to you in your exam, right? But I am going to give you what the cytotoxic T cell is. Now, the cytotoxic T cell is the killer T cell. They directly attack other cells, and the activated cells circulate in the blood and the lymphoid organs in search of body cells displaying antigen that they recognize. Activated T cells would target virus-infected cells, cells with intracellular bacteria or parasites, and cancer cells, and foreign cells, as in when you're experiencing it from transfusion or transplant. Your cytotoxic T cells actually do it in this manner. Now, these are really very complex explanation, but in a nutshell, what they do is that they poke a hole on the cell membrane of the bacteria, and then they release perforins and gran enzymes. Okay, it is just like a soldier opening a window, which is already a hole, and then putting a grenade inside the building to make it explode because there are enemies. So here the T cells would poke a hole on the cell surface, and then they will release perforins and gran enzymes, okay, that would pretty much stimulate the apoptosis of the cell involved. Then you have the natural killer cells. Natural killer cells recognize other signs of abnormality that other cytotoxic T cells do not look for. For instance, the natural killer cells would look for cells that lack class 1 MHC protein. They would look for antibodies coating target cells and different surface markers seen on the stress cell. Natural killer cells use the same mechanisms as uh, uh, cytotoxic T cells for killing other targets. They use also perforins and granzymes, but they are actually the ones that are doing immune surveillance of self cells that has gone bad. So they look for markers that they can recognize in self cells. And for instance, in cancer cells and tumor cells, this is actually the natural killer cells that kill them. Then you have the regulatory T cells. The regulatory T cells are the ones that would dampen or pretty much like end an immune response whenever the immune response is already like uh, closing into an end. Whenever you don't have any more any bacteria, then all of your T cells need to be pretty much like suppressed. Okay, they cannot keep on doing their work if their work is not anymore needed. So they suppress reactive lymphocytes that are in the area that's outside of the lymph organ. Um, Pretty much, there's a big research going on now on regulatory T cells um, because they might pretty much like be good in possibly um, preventing uh, rejection of transplanted tissue. So here is your different types of T cells and the function, and this is a good cheat sheet when you're studying for your exam. Right? I am not. I am pretty much like going to leave this to you to study the transplants and prevention of rejection. And please read on the